Hey everyone, it's Carlos from Smart Edition Academy. In this video, we'll be going over the HESI A2 Biology practice test. You can take this test with me via the link below for a free HESI A2 practice test. Also, in the links below, you'll find our free HESI study group and our HESI online study course. For this test, I will review each question one by one and explain the right answer. Well, grab your notes and get ready to review some biology with me. Let's dive in. In the taxonomic system, a class is grouped into which level? Order, family, species, or phylum? I'll let you think about it for a second. And if you answer D, you are correct. Phylum. The taxonomic system, a class is grouped into the phylum level. The hierarchical of biological classification is organized as follows: domain, kingdom, phylum, class, order, family, genus, species. Each level represents a progressively more specific grouping of organisms. So a class is a group of related orders, which is in turn a group of related families, and so on. Gets a little bit complicated, but essentially that's what it is. And I'm here to help. So here's a popular mnemonic to help you remember the taxonomic system. Dear King Philip came over for great spaghetti. Just write out each of those words, and each of the first letters will represent the taxonomical system. It's an easy way to memorize this, and memorizing that phrase will help you. So let's go into the next question. A researcher characterizes a polymer that consists of glycol molecules. What does she write in her notes about this polymer? All right, these molecules will form lipid biomolecules. This lipid is capable of storing very little energy. The glycol molecules are covalently bonded together. This polymer will help transmit genetic information in a cell. All right, those are a couple mouthfuls, but think about it for a couple seconds. And if you said C, you are correct. The glycol molecules are covalently bonded together. So let's talk about this one. The researcher would write in her notes that the glycerol molecules are covalently bonded together. Glycerol is a component of lipids, but in this case, it's mentioned that the polymer consists only of the glycerol molecules. Therefore, the statement about lipid biomolecules is not accurate. Glycerol itself does not store a significant amount of energy and is not involved in transmitting genetic information in a cell. The most relevant information about a polymer of glycerol molecules would be the covalent bonding between the glycerol units. So, BC, and let's continue. Question 3, still in the same category, everybody. A researcher characterizes the amino acid chain and structure of a novel substance. What type of substance is the researcher studying? Fat, DNA, enzyme, or sucrose? Another second. The researcher is studying C, enzyme. Enzymes are proteins made up of amino acid chains and have specific structures that enable them to catalyze biochemical reactions. DNA and sucrose are not composed of amino acid chains, and fats are primarily composed of glycerol and fatty acids, not amino acid chains. I hope that makes sense. So this one would be C, enzyme. Next one. A study was performed to evaluate which type of road salt de-iced a road most quickly. What is the independent variable? De-icing time period, road used for de-icing, type of road salt used, amount of road salt used. A little tricky, but just think about it. First, define yourself. What is independent variable? All right, the independent variable in this study is C, type of road salt used. All right, I'm going to explain this to help understand all the components in this question. This is because the researchers are changing the type of road salt to evaluate its effect on de-icing time while keeping the other factors like the road used and the amount of road salt constant. Remember, here's the definition. An independent variable is a variable that is manipulated or controlled by the researcher in an experiment study to observe its effects on the dependent variable, which is the outcome being measured. In this case, the researchers are changing the type of road salt to evaluate its effect on de-icing time, which is the dependent variable, while keeping other factors constant. By changing the type of road salt and observing the resulting de-icing time, the researchers can determine which road salt is most effective at de-icing roads quickly. All right. So, next question. A researcher predicts that a new drug will lower cholesterol in people. What is the statement called? Law, theory, variable, or hypothesis? These are the kind of questions where if you know the definition of each of these, it should be pretty quick to the answer, which is D, hypothesis. A hypothesis is a testable statement that predicts the outcome of an experiment or a study based on prior knowledge, observations, or theories. In this case, the statement predicts that a new drug will have a specific effect, which is lowering cholesterol on a specific population, which are people. This prediction can be tested through controlled experiments or clinical trials, making it a hypothesis. All right, question 6. Which trait is used to distinguish living things from non-living things? Size, genetics, homeostasis, or environment? And the answer is C, homeostasis. Homeostasis is the trait used to distinguish living things from non-living things. Homeostasis refers to the ability of an organism to maintain stable internal environment despite changes in its external conditions. This is essential for survival and proper functioning of living organisms. Non-living organisms do not have the capability to maintain such balance. All right. Well, look at that. I told you to keep your eyes on the categories, and we are in a new category, which is cell structure, function, and type. So let's keep going and keep in mind with this new category. Why did it take many years for the cell theory to be developed? Advancements in microscopy took place slowly. Cells were difficult to isolate for experimental analysis. Researchers believed a cell formed from pre-existing cells. Scientists already proved the cells were essential for life. The main reason it took many years for the cell theory to be developed is that A, advancements in microscopy took place slowly. The early microscopes were not powerful enough to properly visualize cells, and it took time for technological improvements to be made. As microscopy improved, scientists were able to observe cells more clearly, leading to a better understanding of their structure and function. Additionally, cells were difficult to isolate for experimental analysis, which also contributed to the slow development of the cell theory. The other two options, researchers believing a cell formed from pre-existing cells and scientists already proving that a cell were essential for life, are actually components of the cell theory itself, rather than the reason for its slow development. A little tricky there, but if you knew the cell theory, this was an easy question. Next one. A researcher discovers a cell that is less than 0.5 millimeters in diameter. This cell has pili surrounding the cell wall. What does the researcher classify this cell as? Autotroph, eukaryote, heterotroph, or prokaryote? The researcher would classify this cell as D, prokaryote. Prokaryote cells are typically smaller in size, usually between 0.5 to 5 micrometers, and have simple cellular structures when compared to eukaryotic cells. The presence of the cell wall and pili are common features in prokaryotes, specifically bacteria. Pili are hair-like structures on the surface of the cell that help attachment and other functions. Now let's talk about the eukaryote. Eukaryotes have a more complex structure, including a nucleus with a nuclear envelope and membrane-bound organelles. Autotrophs and heterotrophs describe how an organism obtains its energy and nutrients not the cellular structure. Autotrophs produce their own food using energy from the sun or other inorganic sources, while heterotrophs obtain nutrients by consuming other organisms. All right, that was D. Next one. All right, which structure do cells rely on for movement? Flagellum, microtubule, pili, or vesicle? Cells rely on A, flagellum, for movement. Flagella are whip-like structures that extend from the cell membrane and help propel the cell through its environment. Flagella are found in prokaryotic and eukaryotic cells, although their structure and function may differ slightly different between the two types of cells. Question 10. All right, keep it going. What is a characteristic of eukaryotes? These organisms are unicellular. They lack the membrane-bound nucleus. Many organelles are in their cytoplasm. Bacteria and plants are examples of eukaryotes. All right, you should be confident with this answer based on all the other questions that we've had so far. A characteristic of eukaryotes is that C, many organelles in their cytoplasm. Eukaryote cells have a membrane-bound nucleus and other organelles such as mitochondria, Golgi apparatus, and then endoplasmic reticulum, which are also enclosed by the membranes. This distinguishes them from prokaryotes, which lack a membrane-bound nucleus and organelles. Eukaryotic organisms can be unicellular or multicellular, and they include plants, animals, fungi, and protists. Bacteria are not eukaryotes, and they are prokaryotes. All right. That was C. Number 11. What cellular process do autotrophs rely on to obtain energy? Carbon fixation, cell respiration, photosynthesis, or gluconeogenesis? All right, the correct answer is C, photosynthesis to obtain energy. Autotrophs are organisms that can produce their own food from inorganic substances, such as water, carbon dioxide, using external energy sources like sunlight. Photosynthesis is the primary process that most autotrophs, such as plants, algae, and bacteria, rely on to obtain that energy. In photosynthesis, autotrophs use light energy to power a series of chemical reactions that convert water and carbon dioxide into glucose, which is a type of sugar, and other organic molecules. Oxygen is released as a byproduct of these reactions. The glucose and other organic molecules can be then used as energy source for the organism's metabolic processes. Just in case, let's quickly go over the other options just so you understand them as well. Now, the other options are cellular respiration. This process occurs in autotrophs and heterotrophs, organisms that cannot produce their own food and must obtain it from external sources. Chemosynthesis. This process is used by some other bacteria and archaea that produce energy in the absence of sunlight. And gluconeogenesis is a metabolic pathway that allows cells to produce glucose from non-carbohydrate sources. Now, this one was, I'll admit, a bit tough, and I would definitely review the topic and answers if you did not feel comfortable with them. This is one where you're just going to have to understand the definitions of each of these functions. All right, question number 12. How is a plant different from an animal cell? It lacks a cell wall. It is rectangular. It has many vacuoles in the cytoplasm. It traps sunlight using its mitochondria. All right, I'm gonna change this one up a little bit. Let's go through the options first. Do plants lack a cell wall? No. They do have a rigid cell wall. Animal cells just have cell membranes. Are plant cells rectangular? Yes, they are. Animal cells are not. Do they have many vacuoles in the cytoplasm? No. They have a large central vacuole where animal cells have many smaller ones. And finally, do they trap sunlight using mitochondria? Nope. They trap sunlight using chloroplasts. Mitochondria is the powerhouse of the cell. So the answer is B, rectangular. All right, next question. So cellular reproduction, cellular respiration, and photosynthesis. All right. Glycolysis breaks down what? Acetyl-CoA, ATP, glucose, or pyruvate? All right. I kind of talked about it before. So glycolysis breaks down C, glucose. Keyword glycolysis. Glycolysis is a series of biochemical reactions that occurs in the cytoplasm of the cells. It is the first step in cellular respiration and involves the breakdown of one of the glucose molecules, a 6-carbon sugar, into 2 molecules of pyruvate, a 3-carbon compound. This process generates a small amount of ATP, which is the cell's main energy currency, and NADH, which is an electron carrier used in later stages of cellular respiration. Next question. During the G2 phase, more copies of tubulin are made to separate what? Histones, chromosomes, daughter cells, sister chromatins. During the G2 phase of the cell cycle, more copies of tubulin are made to separate B chromosomes. The G2 phase is the second gap phase in the cell cycle, occurring after DNA replication during the S phase and preceding cell division during the M phase. During the G2 phase, the cell prepares for cell division by synthesizing proteins and organelles required for mitosis. One of the key events during mitosis is the separation of duplicated chromosomes into 2 identical daughter cells. This process requires the assembly of spindle apparatus, which is composed of microtubules that attach to the chromosomes and then pull them apart. Tubulin is the protein subunit that makes up those microtubules, so the cell needs to produce more tubulin during the G2 phase to build the spindle apparatus, which separates the chromosomes during mitosis. Histones are the proteins that package the DNA into chromatin, and their role is primarily to regulate gene expression rather than to separate them during cell division. Sister chromatins are 2 identical copies of a single replicated chromosome that are held together by a centromere, and they are separated during mitosis. The separation of sister chromatin also requires spindle apparatus, which is built using tubulin. So I broke it all apart just so you have the understanding of each of the options. Again, I think the best strategy sometimes for these questions is to break it apart, define each of the choices, and then understanding which one is the best one based on that understanding. All right, question 15. Let's keep it going. Which process involves crossing over? Mitosis, meiosis, Calvin cycle, or cell respiration? Crossing over during B, meiosis. The process is responsible for genetic recombination and variation in sexually reproducing organisms. Crossing over specifically during the prophase 1 of meiosis 1, whereas homologous chromosomes exchange genetic material, leading to the production of unique combinations of the alleles in resulting gametes. Mitosis is a type of cell division that results in 2 genetically identical daughter cells, each having the same number of chromosomes as the parent cell. The Calvin cycle is part of photosynthesis that occurs in chloroplasts of plant cells. And cell respiration is a process that occurs in mitochondrial cells to generate energy in the form of ATP from the breakdown of their organic molecules. Really, for this question, it came down to 2 of the choices. As I just said, and I kind of teased it to prep you for this question, it always helps to understand the other options so you can break it down to more reasonable choices. All right, so that was the correct answer was meiosis. Next question. What is the correct order of the stages of cell cycle? G1, S, G2, M, G2, S, G1, M, M, S, G2, G1, S, M, G1, G1. The correct order of the stages of the cell cycle is A, G1, S, G2, M. The cell cycle consists of 4 phases. G1, which is gap phase 1, The cell grows in size and prepares for the DNA replication. This is the phase where the cell spends the most time in. And then S, or the synthesis phase, DNA replication occurs, resulting in the duplication of the cell's genetic material. Then G2, or gap2 phase, the cell continues to grow and prepare for mitosis, the process of cell division. And then finally, M, mitosis phase. The cell undergoes mitosis, which is a process of dividing the duplicated genetic material into two daughter cells. All right, I'll admit this was another toughie. If you don't understand the series of events that happen during the cell cycle, it definitely is a hard one. So make sure you understand each of those phases. Next question. What is made during light reaction phase of photosynthesis? ATP, CO2, H2O, or FADH2? During the light reaction phase of photosynthesis, ATP is produced. This phase also generates NADPH. So both of those are energy-rich molecules that are used in subsequent dark reaction phase, which is the Calvin cycle, to produce the glucose and other organic molecules from CO2. The light reaction phase does not produce CO2, H2O, or FADH2. All right. With that, I like to tell you that this is the final section, genetics and DNA. Whew, we're almost there. Come on, stick with me. Which of the following is a segment of DNA that transmit information from parent to offspring? Centromere, chromatin, chromosome, or gene? If you answer D, genes, you are correct. Genes, which are fundamental units of heredity, are responsible for passing down traits from one generation to the next. They are segments of DNA, which is genetic material found in the nucleus of the cells. DNA is composed of 4 nucleotide bases: adenine, cytosine, guanine, and thymine, A, C, G, T, respectively. These base pairs form the famous double helix structure of DNA. Genes are specific sequences of these nucleotide base codes for the production of proteins, which are essential for the proper functioning of cells and organisms as a whole. When an organism reproduces, its DNA is passed on to its offspring, ensuring that the genetic information is transmitted from one generation to the next. In sexual reproduction, both parents contribute to DNA of the offspring, which results in a unique combination of genes from each parent. Each genetic diversity is crucial for survival and adaptation of a species over time. All right, question number 20. The replication of DNA ends with a twisted strand called base pair, nucleic acid, double helix, or single strand. I think you could probably eliminate one pretty quickly. All right, the replication of DNA ends with a twisted strand called the double helix. The DNA double helix consists of 2 complementary strands that are twisted together, with each strand running in opposite directions. The strands are held together by hydrogen bonds between the nucleotide bases that I mentioned before, forming base pairs, which I mentioned before as well. The double helix structure is essential for the accurate replication and transmission of genetic information during cell division. 21. Be careful with this one. Listen to the wording carefully. Human cells have blank sets of different chromosomes. 12, 23, 46, 50. Human cells have B, 23 sets of different chromosomes. Now, you're probably thinking, why not 46? And that's okay. A lot of people mess this up, including me. This is what makes it very tricky. But the question is asking how human cells have blank sets. So a set is 2, right? So 2 times 23 is 46. So 23 from each of the human cells, right? So just make sure you understand that. So an organism has one of the alleles. All right, next question. How can an organism have two alleles for each trait, but only one of the alleles is expressed? One of the alleles is hidden in the chromosome. One of the alleles is dominant over the other allele. It depends on which allele is obtained from a parent first. The allele closest to the top of the chromosome is expressed. All right, the correct answer is one of the alleles is dominant over the other allele, which is choice B. In organisms, traits are determined by the pairs of alleles, which are different forms of a gene. Each individual inherits two alleles for each trait, one from each parent. The way that these alleles are expressed in the phenotype, which are observable traits, is determined by the principal dominance and recessiveness. A dominant allele is the one that is expressed in the phenotype even when only one copy is present, while a recessive allele is expressed only when two copies of the present or homozygous. If an organism has two different alleles for a trait and one is dominant over the other, the dominant allele will be expressed and the recessive allele will be hidden in the sense that it does not contribute to the observable trait. Question 23, a couple more. An RNA copy of a gene used as a blueprint for a protein is called the mRNA, pre-mRNA, rRNA, or tRNA. The correct answer is A, mRNA, or messenger RNA. Messenger RNA is the RNA copy of a gene that serves as a blueprint of a protein. It carries genetic information from the DNA in the cell's nucleus to the ribosome where the protein synthesis occurs. This process is known as transcription, followed by translation. Pre-mRNA is the precursor to mature mRNA. It is formed during transcription. Pre-mRNA is not the final blueprint for a protein. rRNA, or ribosomal RNA, is a component of the ribosome, which is the cellular machinery responsible for protein synthesis. rRNA provides the structural framework and catalytic functions of the ribosome, allowing to translate mRNA into proteins. rRNA itself is not a blueprint for a protein. tRNA, or transfer RNA, acts as an adapter molecule in the translation process. And again, tRNA is not a blueprint for protein, but rather a crucial component in protein synthesis. Two more, everybody. Two more. We got this. An offspring receives blank alleles for a particular trait from each parent. I think I briefly went over this, so let's see if you remembered. An offspring receives one allele for a particular trait from each parent. In sexually producing organisms, offspring inherit half of their genetic information from each parent. Since genes come in pairs, one from each parent contributes one allele for each particular trait. This means that the offspring will have a total of two alleles from each trait, one from the mother and one from the father. Crossing two heterozygous flowers, big B, little b, crossing big B, little b, what is the chance of obtaining homozygous recessive trait? You need to draw the Punnett square for this to correctly answer this question. So I'm going to break it down, but like I said, you should definitely draw this out. When crossing two heterozygous flowers, big B, little b, crossing big B, little b, you can use a Punnett square to determine the probability of obtaining a homozygous recessive trait. Now, if you drew it out, you would have big B, little b, and then on the side, big B, little b, correct? Now, when you do that, you start filling the squares with the components of each edge. And when you see that, you realize that the Punnett square shows only one result of little b, little b. Now, when you see that, the homozygous recessive little b only occurs once in the four possibilities. Therefore, the chance of obtaining a homozygous recessive trait is 1 out of 4 possibilities, which comes out to be A, 25%. I hope I didn't make that more complicated than it is, but essentially drawing out a Punnett square will give you the best way to answer this type of question. And with that, wow, take a deep breath. We did it. That was amazing. I sincerely appreciate you so much for sticking through with me through this test. Just relax, shake it out again. We are done. So with that, for more HESI study materials, check out the links in the description below for the free HESI practice tests, HESI study group, and the Smart Edition Academy HESI Aligned study course. If this video helped you and you learned some important refresher information on biology while watching, be sure to give this video a like. Again, great job. I appreciate you all sticking with me through this. I hope you enjoyed this and found this very helpful. This is Carlos from Smart Edition Academy. Take care.